If you care anything about fashion, then you'll be the first to jump on the all new craze designer babies. Hello, and welcome to the show about fun facts and a little bit of science. Today, we are talking about CRISPR. Imagine a world where you could walk into a Best Buy to buy a PlayStation. Well, that alone is already hard to imagine. But stay with me. You grab the latest PS20, and on your way to checkout, you notice a display in the aisle. It says, "You no longer have to play in a fictional world to have superpowers. Now you can live it." The all new strength enhancer from CRISPR. Well, it might not be as crazy as it sounds. While we do have some time before we can easily upgrade ourselves like some sort of cyberpunk world, we are already making massive leaps in this area of science. CRISPR, which stands for Clusters of Regularly Interspaced Short Palindromic Repeats, is a tool for editing genomes. What it does is it allows researchers to alter DNA sequences and modify gene functions. It has the possibility to eliminate things like HIV and cancer from the human body. The craziest part is that it can not only do this in someone who currently has a disease, but it can be used to pass along modified DNA to offspring, meaning children could be born with an advanced immune system that prevents them from ever having the possibility of getting that disease. If you're enjoying this episode, be sure to hit that like button and subscribe. And I'll see you next time. The technology actually comes from studying bacteria and archaea. They have a natural defense mechanism that allows them to use RNA to attack invading viruses. When researchers transfer this component into other organisms, like mice or humans, it allows for editing of genes. In 2019, there were 3 patients that went through clinical trials involving CRISPR in hopes to find a treatment for cancer. The patients had a form of cancer that was untreatable by any current method, and even though CRISPR wasn't a miracle treatment, it was able to show very promising results. It was a part of a phase 1 trial, which does not measure effectiveness, but rather safety. In this trial, there were no major side effects, and the cells were able to last up to 9 months in their bodies. And no major side effects is a huge leap forward. The traditional cancer treatments that boost up cells can cause high fevers, low blood pressure, seizures, and more. Basically, what happens is CRISPR works in 2 parts. First, it uses a guide RNA to transport a DNA cutting enzyme called Cas9. Once it reaches a specific area of DNA, it then cuts and replaces the bad sections, altering the genes. In the case of this cancer treatment, they altered three genes in immune cells called T cells. The hope was to increase the efficiency of T cells to kill cancer. The phase 1 trial went really well, but it wasn't perfect. More than 90% of the cuts in the DNA were right on target, but some cuts were off and unexpected. When using the guide RNA to make those cuts, the worst one was able to accurately make nearly 8,000 cuts and only mess up about 38. Those mistakes were not harmful in any way based on their research, but it is certainly possible that a cut could be made where it accidentally promotes cancer growth. In this specific trial, the researchers were using versions of CRISPR from 2016, and several updates have already been made to increase their accuracy. With clinical trials happening all over the world and more often, we are already seeing more promise in the treatment of diseases. Vertex Pharmaceuticals in Boston and London has had great results for people with blood disorders, sickle cell disease, or beta thalassemia. They are able to restart the body's production of fetal hemoglobin, something that is usually made only until shortly after birth. Using CRISPR could mean these patients go in for a one-time treatment. Vertex also used CRISPR to lower cholesterol in monkeys by 60% in just 1 week, and the levels stayed low for over 8 months after a single dose of the treatment. Then there's the trial where researchers are working to restore eyesight in people who inherited Leber congenital amaurosis 10. They are able to cut out the part of the DNA that causes the mutation responsible for this blindness. Last week, I talked about genetically modified mosquitoes. Well, there are companies that are using CRISPR to do the same thing, help stop the transmission of diseases from specific species of mosquitoes. A link to that video is in the description down below. But what about those designer babies I mentioned at the beginning? Well, yeah, that's a topic of concern for some people. In what is called germline editing, researchers are able to correct disease causing genes. It's not intended to add any new genes or alter traits. For example, in 2017, CRISPR was used to help remove a genetic defect that is known to cause heart failure. Researchers were working to remove the mutation known as MYBPC3, which causes sudden heart failure. It's an inherited trait that hits about 1 in 500 people worldwide, but there is no cure, only treatments for symptoms. So finding a way to correct this is pretty important. In this study, researchers were able to inject that Cas9 enzyme we mentioned earlier into the egg of female patients along with the male sperm that contained the mutation and a healthy chunk of DNA. The hope was that Cas9 would replace the mutation using this added DNA, but what they found was Cas9 was using the healthy genes of the mother and not the extra DNA they sent in. This was a surprise, but an exciting surprise. The unknown factor now is what would happen if both the male and female donors had the inherited mutation. Would the added DNA sent in with Cas9 work in that scenario, or be overridden again by the female genes? This new finding could be good information for anyone concerned about designer babies. It might be impossible to add or alter traits in that way. The great thing here is that there were no signs of Cas9 cutting the wrong strips of DNA as shown in the past. It's possible that inserting Cas9 along with the sperm as it enters the egg, and not after, will help resolve any mistakes. While it may be unclear for now if we are able to add or alter genes to make super babies, it might be worth the journey in the meantime to help save lives. This is why we have regulations, but it just takes one mad scientist to make that first jump down the rabbit hole. For now, everything is in the hands of these researchers and the US government. With great results from clinical trials, it might satisfy the right leaders to eventually be used in everyday medicine. Who knows what will come next? Do you think we should be messing with human DNA in this way, or is this just asking for trouble? As always, thanks for watching, and what did you learn today?